Okay, so let me call it chapter 3. In chapter 3, we're going to talk about biologically important molecules. We talked about one, which is water, but water is a pretty simple molecule. Here we're going to shift our attention to things called macromolecules. Okay. So, within the context of biology, mostly we're interested in things called organic molecules. And organic molecules consist of 3 primary elements, and those are carbon, hydrogen, and oxygen. The vast majority of the molecules that we're going to be dealing with deal or consist exclusively of carbon, hydrogen, and oxygen. Examples of biologically important molecules that we're going to talk about are carbohydrates, which are our sugars, lipids, which are the fats, proteins, and nucleic acids. And nucleic acids are our DNA and our RNA. Now these are all fall into the same class of macromolecules. And a macromolecule is a large complex molecule that commonly consists of repeating subunits. Those repeating subunits are called monomers. So for example, we're familiar with the molecule starch, right? We get starch out of potatoes and corn and rice and wheat and things like that. And really, starch is just a long chain of molecules called glucose. And glucose, as you perhaps know, is just a sugar. It just continues on like that. This is a macromolecule. It's a large complex molecule. The individual subunits of the starch molecule, the monomers, are glucose. If you take all of those monomers and you stick them together, you create a polymer, which is that macromolecule. Another good example are proteins. Proteins are just repeating amino acids. So if you take a whole bunch of amino acids and you stick them together, there's a covalent bond, then they form a polymer of those monomers, which is a protein. Now we use a couple of reactions to stick monomers together. There's something called dehydration synthesis, or a dehydration reaction, and then there's something called hydrolysis, or a hydrolytic reaction. With dehydration synthesis, or dehydration reactions, we're taking 2 monomers, removing a water molecule from them, and sticking them together. Let me show you how that looks really quickly. We'll use glucose again. So glucose, let me draw out the entire molecular structure of glucose. Glucose consists of 6 carbons, 5 of which exist in a single ring. And then a 6th is up here. And we have this attached. So that's what glucose looks like. Now if we wanted to take this glucose and we wanted to stick it to another glucose molecule that looks exactly the same, I know it's super interesting watching you draw. We could use this process called dehydration synthesis to connect those 2 glucose molecules together. Now, with dehydration synthesis, what we're going to want to do is to take a water molecule out of these 2 molecules collectively. We're going to need to take 2 hydrogens and an oxygen from those 2 molecules. So what we do is we take this OH off of this molecule, and we take this hydrogen off of this molecule, and we stick them together, so that we get H2O. What that's done is it's freed up a bond for this carbon right here, and for that oxygen right there. So that at the end, what this side and that side look like is this. So that now this carbon is attached to that oxygen, and that oxygen is still attached to that carbon, and those two molecules are stuck together. Now again, that's called dehydration synthesis or dehydration reaction, because we've pulled collectively a whole water molecule off of those two individual monomers. We can do the opposite, and the opposite is called hydrolysis or a hydrolytic reaction. Hydrolysis is taking these two glucose molecules that are stuck together, inserting a water in there, breaking those bonds, and separating the two monomers. You guys, at night, when we're trying to replenish all of our energy in our muscles that we've expended, we do a lot of dehydration synthesis. Our muscles store a large complex sugar called glycogen. And glycogen is just a long series of chains of glucose molecules. And so when we're resting, we're metabolizing everything that we've eaten, and we're taking those glucose molecules, transporting them to our muscles, and dehydration synthesizing large glycogen molecules. Okay? During the day, when we're being active, we're doing the opposite. We're doing a bunch of hydrolysis. We're taking off individual glucose molecules and burning them up so that our cells have some energy to do work. Now, commonly, organic molecules are going to have these kinds of side groups. For example, an amino group is a really important part of our amino acids. So in addition to carbons, hydrogens, and oxygens, proteins also have a lot of nitrogen in them. And then you'll see groups like this, like the carbonyl group or a hydroxyl group. This is another really interesting and important one, and that's a large phosphate group. And that'll be important for our nucleic acids. Okay, so this is again what dehydration synthesis looks like. Dehydration synthesis is where we can take two monomers, remove a water from them, freeing up a bond right here and connecting those two monomers together. Now, this indicates that energy is invested into that reaction, and that's true. Whenever we're making something more complex, attaching two things together, it's going to require an investment of energy. Every time we break bonds, however, we're releasing energy. So this is an example of hydrolysis, where we're taking two monomers and separating them with water so that we now have two molecules instead of one large complex molecule. And when we break that down, we release a little bit of energy. Okay, so we're going to talk about four biologically important molecules. What I'm going to want you to know for each of these molecules is what their basic structure is and what their function is. And by structure, really what I mean is, what are the monomers and how do we connect them together? Each of these molecules is going to have a unique type of bond that connects monomers, and I'm going to want you to know that bond. Okay? So, carbohydrates. The real function, particularly within animals, of carbohydrates is for energy storage. And it's primarily short-term energy storage. Large polymers of carbohydrates are pretty clunky molecules. They're really large, they occupy a lot of space, and they don't store a ton of energy. And so really, it's for immediate use. If we want to store energy for long-term purposes, then what we need to do is develop a molecule that stores a whole bunch of energy in a very small space. And we're going to talk about that class of molecules next. But carbohydrates, these are for immediate use. So generally what happens, right, is when we're starting to do something aerobic, our body has some priority in which it's going to burn energy. We know that we can store energy as carbs, we can also store energy as fats. Because carbs are so much more accessible, so much larger and so much clunkier, our body prioritizes those. It's going to burn up our carbohydrates first. Okay. So within the carbohydrates, there are really three classes of carbohydrates that I'm going to want you to be familiar with. The first of which are called monosaccharides. And monosaccharides are all unified by a similar chemical formula. And their chemical formula is CH2O. What that means is, within monosaccharides, for every one carbon, there are 2 hydrogens and 1 oxygen. Glucose, if you were to count all of the oxygens, hydrogens, and carbons up, would have the chemical formula of C6H12O6. Like that. There's another monosaccharide called fructose. And fructose has the exact same chemical formula, C6H12O6. There's a sugar that all of us use in our DNA, and it's called deoxyribose. Deoxyribose is a 5-carbon sugar called a pentose. So if it truly is a monosaccharide, how many hydrogens should deoxyribose or ribose have? 10, exactly right. And oxygens, it should have 5. Very good. There are also 3-carbon sugars called trioses. So if there are 3 carbons, there have to be 6 hydrogens and 3 oxygens. So all monosaccharides follow that general chemical formula. For every 1 carbon, there are 2 hydrogens and 1 oxygen. Monosaccharides are interesting and important because these represent the foundation of our metabolism. When our cells are burning energy, largely they're doing it in the form of monosaccharides, specifically or hexoses, sugars like glucose and fructose. But what we can do is we can take 2 monosaccharides and we can stick them together with dehydration synthesis and make a disaccharide. So, good examples of disaccharides are sucrose. Do you guys know what sucrose is? Yeah, it's just table sugar, right? It's the kind of sugar you put in your coffee or in your tea or on your cereal in the mornings. It's estimated that the average American consumes 150 pounds of sucrose each year, which is crazy. It's a phenomenal amount of sucrose. Another really good example of a disaccharide is lactose. And what is lactose? Yeah, it's milk sugar. And milk sugar is interesting because it's a disaccharide of glucose and galactose. Now you'll notice that glucose and galactose are very, very similar. The only difference is right here, right? So in glucose, the OH group, this hydroxyl group is on the bottom, and here that hydroxyl group is on the top. So, what does lactose intolerance mean? Is there anybody in here lactose intolerant? I shouldn't smile, but yeah, it's sad. You guys can't eat yummy things like ice cream and cheese without taking lactate, right? So what does lactose intolerance actually mean?
You're intolerant to that sugar.
And intolerance means what?
Like a word.
Yeah, you can't break it down. And so when you guys who are lactose intolerant consume milk, that sugar just remains in your intestine. And then you get a secondary bacterial growth on that milk sugar, and then it causes bloating and gas and all sorts of discomfort, right? The reason that individuals are lactose intolerant is because there is an enzyme that recognizes the bond between glucose and galactose, and lactose intolerant individuals don't manufacture that enzyme. Lactate is an artificial enzyme that breaks down lactose to make it glucose and galactose. And so if you were to take lactate and you were to dump that into your milk, the milk is actually going to taste sweeter. Even though you haven't changed like the amount of sugar in it, it's going to taste sweeter because you made a disaccharide monosaccharides, and monosaccharides just taste sweeter than disaccharides do. Anyways, individuals that are lactose intolerant lack that enzyme called lactase, so they can't break down the bond between glucose and galactose, but they are capable of breaking down glycogen, which is two glucose molecules attached together. So this one minor shift in the relative position of that hydroxyl group creates a bond between two molecules that are just a little bit different. And that minor difference makes the enzyme that can break down two glucose molecules ineffective against glucose and galactose. It's interesting because, and you guys will read a little paper about this as well in lab, but there are specific ethnicities where lactose intolerance is much more common. Individuals of Asian descent have a much higher occurrence of lactose intolerance than those that are Anglo. Same thing with African descent and Latino descent. Yeah.
Is it like a genetic thing? Like
it
if your
is.
parents have it, then you're like more likely to have it?
It is, and it's really bizarre because really our ability to digest milk sugars is a product of whether or not our ancestry had cattle in agriculture for a long time. So the Anglo-Saxon descent, we've had cattle in agriculture for a really long time. Those that haven't typically don't drink milk into their, well, like later years, right? And so there wasn't that need for lactase after you get a little bit older. And so individuals whose ancestry doesn't have a history of cattle in agriculture have a much higher occurrence of lactose intolerance. Okay, so, oh, sorry, let me mention one more thing. With carbohydrates, the bond between monomers, when we make like a disaccharide, that's